Welcome to our channel. In this video, we are going to see about GPCR pathway, G protein coupled receptor signal transduction pathway. So in this video, we are going to see about the structure of the GPCR and the mechanism of this pathway. So if you want to see about the general information about the cell signaling, I have given already about the cell signaling pathway in our channel. You just watch it. G protein coupled receptors are a diverse family of receptors. They are found in a huge range of tissues throughout the body. They function to respond to a wide variety of extracellular signals. Actually, when reacted with some reactions, our body reacts with some signaling. So GPCR signaling is one of the very important pathway. It is actually activated by some hormones or neurotransmitters and trigger intracellular signaling cascades which regulate a wide range of the body functions. Structure of GPCRs. G protein coupled receptors are composed of transmembrane region. It crossing the lipid bilayer 7 times. So this is called 7 transmembrane receptors. This region is coupled with the G protein. GPCRs have no integral enzyme activity or ion channel. Therefore, all their downstream effects are mediated via their G protein only. The G protein actually have the heterotrimeric structure. Means it made up of 3 different subunits like alpha, beta and gamma subunits. So in the inactivate state, GDP is bound to the alpha subunit of the G protein. You just to see the picture about this GPCR, like see, this is alpha unit, beta unit and gamma unit, and this is the structure, 7 membrane structure, and this is the extracellular space, and this is membrane. So exactly what is the structure of this GPCR? 7 membrane structure, alpha, beta and gamma unit. In the inactive state, actually this alpha unit binds with the GDP. You just see the picture here. But in the activated state, actually alpha it is bind with the GTP. The GTP is converted as a GTP. So it involved in the cellular response. There are hundreds of GPCRs in the genome, and their receptors are activated by many signals such as I already said that neurotransmitters, hormones, ions, peptides and even photons in the retina. Common examples of GPCRs are include adenoreceptors and muscarinic acetylcholine receptors and opioid receptors. There are also many different types of alpha, beta and gamma subunits. This allows for many GPCR combinations created by different receptors. It being coupled with G proteins, comprised of different subunits. Furthermore, one GPCR can be associated with many G proteins. Hence, one signal can have many downstream cellular reactions. So this is a very important point. One signal actually have many downstream cellular reactions. So this is steps about how this GPCR is activated, what is the mechanism and what is called the ligand. Once ligand binding with the GPCRs means that the receptors, how the cells are reacted. So this is about the mechanism of this GPCRs. And agonist means ligand as a substance is which binds to a receptor and brings about the cellular responses. So in the signal transduction pathway, actually 3 stages. One is the reception and signal transduction. Last one is cellular response in general. So the G protein activated only when the ligand binds with this particular particle. For G protein coupled receptors, this consists of 5 main steps. So first step. So the first step is the ligands bind to the extracellular portion of the G protein coupled receptor, means the outer area of that receptor. It binding either at the N terminus or a binding site within the transmembrane region. See the picture. So first one is inactivated stage, the receptor inactivated stage. One it's bind with the ligand. It is activated. See the. So this is inactivated stage. The structure actually have alpha, beta, gamma, and here the alpha is bind with the GDP. So this is inactivated stage. Once the ligand bind with this membrane, and this is activated stage. See that when it gets signal, it is activated. The GDP is converted to GTP. When it is added with the phosphate, GDP converted to GTP. So once it is GTP binded with the alpha in it, it dissociates from the membrane and it is involved in the cellular responses. So the release the GDP is then replaced with the GTP. This activates the G protein, it causing the alpha subunit and bound GTP to dissociate from the transmembrane. So automatically when it is formed as a GTP, the alpha plus GTP dissociate from the transmembrane portion of the GPCR and from the beta and gamma subunit. This alpha subunit interact with their relevant effectors. So it causes many downstream effects, not only a single effect, it causes many downstream effects like ion channel opening or enzyme activity regulation, like that. Despite the fact that one G protein coupled receptors only contains one alpha subunit, but this can interact with several secondary messengers which can in turn activate multiple enzymes and catalyze many reactions. This creates a cascade response whereby one agonist means ligand in binding to the GPCR can bring about the catalysis of many reactions. Actually this is called signal amplification. Only one single alpha unit, but it interacts with many secondary messengers. It can activate many enzymes and also involved in many reactions. So that's why it is called signal amplification. To prevent the excess signaling, the GPCR activity can be even though switched off. How it is possible means by the enzyme GTPase. This GTPase, it catalyzes the breakdown of GTP on the alpha subunit into GDP plus phosphate. That means the reverse reaction. So once GDP binds with the phosphate, it forms GTP and it is dissociated from the membrane. So if it is the mechanism switched off means again it will form the GDP. So GTP to GDP. So it is switched off condition. It allowing the reformation of the heterotrimeric complex of the G protein. The alpha, beta, gamma associated with the membrane protein. So the G protein then reassociate with the transmembrane receptors. So it reforming the GPCR for the next ligand binding. So what are the types of G protein? There are several different types of G protein that can be present in a GPCR. So I already said that only one alpha subunit, but it involved in many downstream processes and it involved in many reactions. So it is called signal amplification, but it is vary based on the alpha subunit. Each alpha subunit stimulates an enzyme which acts to either increases or decreases the concentration of the secondary messenger. So depend upon the reaction change here. This goes on to impact a downstream effector which then causes a cellular response. The ultimate effect of these proteins depends on the specific cell in which it is located. So depend on where it is located and where position it is located. See, I have given some example. Only a single alpha subunit, but it have many subunits. GS. It creates an enzyme, stimulates adenyl cyclase, which catalyzes the conversion of ATP to cyclic AMP. What is the secondary messenger formed here means it increases AMP. CAMP is one of the secondary messenger. Effector, it stimulates PK activation. CAMP dependent protein kinase, which goes on to the phosphorylate target proteins. Next one is GI. So this alpha subunit actually has opposite effect for GS. Means GS stimulate adenyl cyclase, but it inhibit adenyl cyclase. That is a major difference, which catalyzes the conversion of ATP to cyclic AMP. So the enzyme is same, but it inhibits adenyl cyclase, reduces CAMP, because in the first it increases CAMP, GS, GI reduces CAMP. So automatically the reaction is have the opposite reaction. And third one is GQ or G11. It stimulates phospholipase C, which cleaves PAP to the cell membrane into IP3 and DAG. It increases IP3 and DAG. IP3 opens actually calcium channels, it causing a calcium efflux into the cytoplasm. So it has some cellular mechanism depend upon the subunit only. Not a single alpha involved in same reaction. It have many subunits that involved in many reactions, cellular responses here. You see the picture here. How the whole mechanism involved, like how this receptors bind with the ligand, and how this activator and various forms of subunits formed here, then involved in many cellular responses. So this is the overall reaction about this pathway. Thank you for watching.